Hey, and today I'll be explaining protein synthesis. To begin with, this is Gary, and he knows everything about protein synthesis, and so will you soon. Every one of our cells, even Gary, is made up of these tiny little building blocks called cells. If we look closely, our cells are mainly made out of two things, water and proteins. Proteins! How are they made? Gary, I know you know, but just be quiet. Before we begin, you need to remember three objects inside the cell. The cytoplasm, the light blue thing you see, it's kind of the open space of the cell, packed with cell highways and houses. Then the ribosome, the black small dots you see, and then the nucleus, the big black dot in the center. You don't need to know the rest of the organelles for this video. The making of proteins begins in the nucleus. There, we have this wonderful thing called the deoxyribonucleic acid, or DNA, and little sections of it, called genes, code for different proteins. The DNA codes using the four bases, called A, T, C, and G, or adenine, thymine, cytosine, and guanine. There's a specific rule that A always pairs with T, and C always pairs with G, otherwise you might have a genetic disorder. So basically, the bond between the base pairs splits for a certain gene, and then bits of RNA pair up with the DNA to make this thing called messenger RNA or mRNA, because DNA is just way too fat to fit in the nuclear pores. RNA isn't friends with the base called T, therefore it replaces T with U or uracil. Gary, don't you dare fall asleep. When DNA makes RNA, it's called transcription. Then mRNA travels out of the nucleus through the holes in the nucleus called nuclear pores and out into the cytoplasm. Then it reaches the ribosome, the protein-making machine. The ribosome grabs onto it and starts reading it. So you know the bases on mRNA? Well, every three of them in a row are called codons. Each combination codes for a specific amino acid, the building blocks of proteins. There are 20 in total, actually. So there is this thing called transfer RNA, or basically tRNA, which looks something like this, with an anticodon there and an amino acid there. However, for the purposes of this video, tRNA will look like this. Did I mention the ribosome is also RNA, rRNA, or ribosomal RNA, which actually looks like this. So, a tRNA pairs with mRNA inside the rRNA, matching codons with anticodons, and soon we have a chain of amino acids, a peptide chain.
Because the bonds between them are called peptide bonds.
This whole process is called translation. The peptide chain then folds to become a protein, which is then let out into the cytoplasm. Cool, isn't it? So guys, I hope you enjoyed my first ever video on Geek Overdose.